Welcome to my art piece and audio snippet inspired by the book Spineless, The Science of Jellyfish and the Art of Growing a Backbone by Julie Burwald. My favorite chapter was Chapter 8, entitled Day Glow Jellies. This chapter describes the discovery, isolation, and purification of fluorescent and bioluminescent molecules from the jellyfish species Eichoria victoria, also known as the crystal jelly. It focuses on the researcher Osamu Shimomura, who solved the mystery of the unidentified green glow that crystal jellies emit. It took 5 years of intensive research for Shimomura and his team to identify the protein luciferin that releases a blue photon when interacting with the eichorin protein and calcium molecules. This discovery was just the first step, however, because crystal jellies actually glow green in the wild, not blue. The blue light made by luciferin is actually turned into the visible green light by green fluorescent protein, or GFP. GFP is now an extremely important and widely used molecule in scientific research labs, from cancer and medical research to plant genetics and worm neurobiology. GFP glows green under blue light automatically without enzymatic assistance. Although crystal jellies are not endangered, this research ended up killing thousands and thousands of them to extract their luciferin. Shimomura and other researchers would catch them, cut off the edge of their bell, and squeeze them out into containers to bring to the lab. They ended up using 2.5 tons of these jellyfish just to get 1 milligram of the molecule luciferin to characterize it. I worked closely with GFP in muscle cell research last spring, and I have a ton of images of green fluorescing muscle cells under a fluorescent microscope. That's where this image came from. What you're seeing on the screen are rat muscle cells that I genetically engineered to produce GFP. This is how I identified living cells in my culture. This photo in particular is a bit overexposed, but that is so that I can appreciate the bright green color. After being inspired by this book, and in particular all the information about GFP and its discovery, I found images of crystal jellies online and I layered them onto my research image. I wanted to have them very faint, as if I was capturing the ghosts of these crystal jellies who were unwillingly sacrificed for GFP to be able to exist. After reading this book, I feel like crystal jellies aren't given enough credit for GFP. I always knew that GFP came from jellyfish, but I didn't know which one specifically, and I didn't know how it was discovered before reading this book. Since I have been thinking about non-human research ethics in a new way this semester, this book has reminded me of the story of Henrietta Lacks. She unwillingly and unknowingly made a major contribution to research and modern medicine. She saved lives and was treated wildly unjustly. By no means am I trying to compare Henrietta's story to that of the jellyfish, but I wanted to make that connection so I could frame the non-human jellyfish as being just as important as humans. Non-humans are so often seen as less than humans due to our human exceptionalism ideologies. GFP from the crystal jellies is a huge discovery, and its uses in research have also contributed to life-saving medical discoveries. Green fluorescent protein is so great because it allows us to see things that we could never see before. By attaching a GFP gene to a protein gene, we can use fluorescent microscopy to see what cells have made that protein. Sometimes we can even see the exact part of the cell where that protein and the GFP are localized. The discovery of GFP is so important that it is comparable to the discovery of the microscope. Non-humans have contributed more to modern science than we realize, and more than we are willing to attribute to them. We take advantage of everything we can under the excuse of understanding and learning about the world and making it better for humans. We do think of animals sometimes, especially when they're at risk of extinction or when their survival and well-being will benefit humans as well. I really wish that all scientists could think about non-humans with appreciation and curiosity instead of something to exploit. Non-humans contribute majorly to science, and they do a lot of labor for us. Examples of this would include lab rats in a medical lab and bacterial colonies in microbiology labs. We need more bioethicists, and we need to figure out how we can balance research advancement and non-human ethics. These crystal jellies contributed to a Nobel Prize, and yet we as humans are polluting their homes.